Okay, we will go ahead and get started. Good morning, good afternoon, good evening. Welcome back to day number 8 of May Institute on Computation and Statistics for Mass Spectrometry and Proteomics. We are very excited about the program today. Maybe before we start, if you are joining us for the first time, some logistics. So if you are joining us through Zoom, at the bottom of your screen, you have two buttons. So one is chat, and you can use this chat button to talk to the panelists or to all the participants. But we prefer that you use it for technical reasons, such as if you cannot hear us or you need a reminder of the URL with materials or something like that. On the other hand, if you have a question regarding the actual technical part of the presentation, please use the Q&A button on the right, because this button alerts us about the questions in real time and allows us to manage the questions more effectively. This said, please don't hesitate to ask questions at any time. So we have a very exciting program for today. We will have two parts of the conversation. The first part focuses on MassIVE.quant. It is a repository for storing, processing, reanalyzing data from quantitative mass spectrometry-based proteomic experiments. And then the second part, we will talk about an extension to MSStats, MSStats-TMT, which is designed specifically for work with TMT labeled data. So without further ado, let me introduce our first speaker, Nuno Bandeira. So Nuno is a faculty at University of California, San Diego, with a joint appointment in School of Pharmacy and in computer science. So Nuno is a computer scientist by training. He has a PhD in computer science from UCSD as well. And he is well known for his contributions of algorithms for analysis of mass spectrometry data. And also Nuno is the person behind MassIVE. So this is one of the repositories for analyzing and storing and analyzing mass spectrometry data. And so I'm very excited to hear Nuno's presentation today about MassIVE.quant, so how MassIVE is extended for quantitative experiments. So thank you very much, Nuno, and welcome to May Institute.
Thank you, Olga, for the kind introduction. Welcome, everyone, to this session. Glad that you could make it here, and glad to have this opportunity to tell you a little bit about what we're doing at MassIVE to work with public proteomics data and to enable not just the sharing, but also the reanalysis and reusage of that proteomics data.
Nuno, we can see you, so maybe add the video.
That's on over here. Okay.
Perfect.
No, thank you. So data sharing in proteomics, you've probably heard of the Proteome Exchange Consortium. If for no other reason, when submitting your papers for publication, the editor would have given you that potentially annoying note at the end that tells you, oh, and by the way, you actually should share your data somewhere and get an identifier for that data set. So the Proteome Exchange Consortium is the network of repositories at a global scale that enables this service. And there's a few of us. There's MassIVE in the US, there's Pride in the United Kingdom, there's iProx in China, JPost in Japan, and others for targeted proteomics data. So it's definitely grown over the past couple of years. And this is a slide that is at this point historical, starting in July of 2016, when at that time there were only 4,500 public data sets. And as you can see, last year at the May Institute, there were about 8,000 public data sets in May of 2019. And today, or actually yesterday, we had over 1,100 public data sets, which is a growth of 40% in just 1 year. So definitely we're seeing a lot of data being shared, and the community has definitely accepted the idea that this is the way to go forward with data in mass spectrometry, is that we'll share it and we'll all be able to access it. However, we're still in a situation where most people think of repositories as something like this, that at the time right before you go to publish your paper, you have to put your data somewhere and you upload upload it there and you essentially forget about it. So it's kind of a view of repositories where it's kind of a data cemetery. It just goes there to never be seen again, other than having that identifier that allows you to get the publication. And one particular thing that I would highlight about this first is just in terms of volume. As I mentioned, we're past the 10,000 data set mark, hundreds of terabytes of data, billions of spectra available in the repositories. But actually, very little of that data is usually uploaded with any kind of identifications on them. So it's not even possible to tell easily what proteins are in each data set. And overwhelmingly, there's pretty much no reusable quantification data, let alone differential analysis of the data that can be reused in the data sets as they're submitted. So to deal with this, and understanding that this was what we would expect from the data, at MassIVE, we have taken a different view of repositories where it's not just a place where you go to deposit data and just leave it there. MassIVE is designed from the start to enable reanalysis of the data. So for one, you will find data sets in MassIVE that might not have been originally deposited in MassIVE because we aggregate data sets from across the whole consortium. Then the data gets converted to open formats. The raw files get converted to MZML, the results get converted to MZTab, and all of those are reanalyzed. Well, not all, but many of those up to this date. They're reanalyzed with advanced search workflows that do not just the original types of analyses, most of the times, just most protein identification. Here we look for modifications, we do proteogenomics, we do other kinds of searches. And then we have advanced visualization interfaces that let you explore the data and interact with the data beyond just downloading data sets. So it's really designed to be a more dynamic kind of environment. And that's why MassIVE is actually an acronym for the Mass Spectrometry Interactive Virtual Environment, where you can interact with the data without having to download it and redo all the hard work of reanalyzing it yourself. So if you go to MassIVE, of course, at the start of the homepage of MassIVE, the first things that you'll see, beyond just some statistics of what's happening in a repository on that day, where we are with numbers, you'll have the two common things from repositories. You can access the public data sets, or you can submit new data sets, where it's also where you find the ability to do other things, such as reanalyzing, comparing, and that's what I would like to emphasize a little bit more today, although of course I'd be glad to talk more with any of you if you're interested in understanding how to submit data to MassIVE. But today we're really focusing more on reanalysis. So let me get to what a dataset page looks like. So if you come to MassIVE, a dataset page would look somewhat like this. And I'm going to highlight a few things on these pages. The first thing that I'll highlight here is how MassIVE connects different resources. So for this particular example, this is actually a CPTAC dataset that is originally deposited at the NCI Proteomics Data Commons, but was deposited in MassIVE to then get a Proteome Exchange identifier, which is now linked to Proteome Exchange, as you can see through the identifier here, while also being linked back to the original source of the data where it came from. Now, if you deposit data at MassIVE and you own the data set, you'll see these buttons next at the top of the dataset page for each data set that allows you to add files, add or update metadata, or add publications to your data set. And this is one way in which data sets at MassIVE are dynamic, in the sense that you can add things to it, you can update it, and you can add your publications, which is important as you keep publishing on the data that you first published maybe a couple of years earlier, but now you're doing new things with it. Even if you don't own the data set, there's a number of things you can do, as you see on the top row. The top row is for anyone that comes to a data set. The bottom row here is only for data set owners. So you'd only see it if you are the person who owns the data set and submitted it. You can convert it. But anyone can come to a data set and convert it to open formats. In this case, there's nearly 20 million spectra in this data set. And you can reanalyze spectra. So these workflows that I'm showing here on this slide are actually already available at MassIVE, and you can run your search on our system, again, without needing to download the data and without needing to reprocess it locally, which in some cases can take some time. So as I mentioned, for this data set, there's 20 million spectra, is 1.5 terabytes. For many of us, just even downloading that and converting it to open formats would already be a pretty significant task. But here, pretty much at the click of a button, you can access all of these workflows for proteogenomics searches, for modification searches, or even data-independent acquisition searches. But again, more important for us today, there are also workflows already in the system that will allow you, for example, to run MaxQuant to do label-free quantification on these data sets. And of course, the jewel at the end, we can run MSStats to do the statistical analysis of the differential expression. And of course, that's what you've been hearing a lot about this week and last week in the previous sessions. And I'm glad to say that this is also one of the options that we have available here. So on the other hand, if you do things offline, you can also add it as a reanalysis to a data set. And in that case, you get assigned your own identifier for your reanalysis, which you can then use for publication. And at this point, MassIVE is actually the only repository that lets you do this, because again, it was designed from the start to be used for reanalysis.
Could you take one question?
I just saw.
Oh, okay.
So, and I was just about to answer, but the question is, how much data can I upload to MassIVE? Is there a maximum memory? I presume that's a volume allocated to each user. The answer is there's currently no maximum. The largest data set that we have that has been deposited is 12 terabytes. So if you do plan to deposit something of that scale, please do contact us first, just to make sure we can help you with the data uploads and make sure everything is good. But we'll definitely be able to take it in. And does the data have to be kept as public? The answer is no, while the ultimate goal is that the data will become public. Of course, there's a period during which you're constructing the data set, you're putting things together, or the papers and the review. Of course, you don't have to have the data be public during those periods. Data sets are by default private. You get a password and reviewer access credentials, and only people with the credentials can access the data set. Until you decide to make it public, which is something that you have to do very explicitly. You have to come to the repository and click a button that says make public, and only then does it get public. So I hope that answered the question. There's no predetermined limit, and it does not have to be public to begin with. So with that, so we can do all of these reanalyses, and in this case, you can see here, there's already 5 different types of reanalysis of this CPTAC data set. So then what happens? How can you work with these reanalyses? Well, the first thing you can do is, if you're interested in comparing 2 specific reanalyses, then there are also workflows at MassIVE that you can use to compare results from different searches. For example, you could compare your own reanalysis results to the dataset results. So as I show here in this example, and here's my pointer, there is a way to select results from the dataset itself, which might have been the original results that were submitted with the dataset. And then you can compare that with your own search results from reanalyzing that data. And you would get a comparison of the 2 sets of results. Where were they the same? Where were they different? And you can see that at the spectrum level, peptide level, and protein level, just to get an idea of things compare. Or if you're interested in having a broader search across all the data that's available on the repository, you can also do that. In this panel that shows in the MassIVE homepage, you can directly search all submitted identifications, and that includes both the originally submitted IDs and the reanalysis IDs. So you can come here, and at the click of a button, you can type up any peptide sequence you're interested in or any protein you're interested in, and then you can search across all the data sets to find where has the protein been seen, where has this peptide been seen. And it searches for you over essentially 500 million spectrum identifications that we have in the system already. Or if you're interested in using it to analyze new data, we have also distilled all of these reanalyses at MassIVE into the MassIVE knowledge base spectral libraries. So as shown in here, this is a human spectral library derived from over 30 terabytes of human MS data. And as far as we know, it's still the largest HCD spectral library for humans. So if that's the kind of data that you're interested in processing and working with, I would recommend you give it at least a try to see if it helps you with getting additional IDs, because it covers over 50% of the human proteome as is. And it includes hundreds of thousands of spectra from synthetic peptides from the Proteome Tools project. So you can also rest confident that these are reliable IDs in that sense. On the other hand, if you're more interested in exploring the data from a protein-centric perspective, I'd recommend using Protein Explorer, as in the entry point there is per protein, and then you can see protein coverage and a lot of other things from the repository scale results, but in a protein-centric view of the world. Coming to the main star for today's topic, MassIVeQuant has really been a great recent development that we've integrated into MassIVE, and that is this global resource that is designed for systematic sharing of quantitative mass spectrometry data. You've run quantitative mass spectrometry experiments yourself, if not before, during the last week and during the sessions this week. So you know there's a lot of steps to take care of. There's a lot of different ways in which you can search the data. You can use different search tools, you can use different quant tools, and you can have different types of mass spectrometry experiments. All of these things requiring different configurations on how you run the software and how you use it. So it's very useful to have a place where you can deposit all the details of these analyses in a manner such that you can go back and reproduce those searches yourself, because that is the first step, or one of the very important steps, in then being able to do it again for your own data going forward. So MassIVeQuant provides a new infrastructure for sharing this kind of information. And to get it started, we already populated it with over 40 different data sets and close to 180 different reanalyses that go all the way from the data, quant analysis, metadata, and MS stats analysis, so that you can see the details of how the results are derived all the way from the start. And this, of course, was developed together with Meena and Olga, who are here with us today, but also with Ben, Julie, and Jeremy, who put in a lot of work to make this happen on the MassIVE side. And I wanted to make sure to not let that go by, because they're really, together with Meena, the ones to thank for having this system put together. So to give you a quick idea, and we'll go into more detail on this for the rest of the session, but just to give you a couple of points on the major new features of MassIVeQuant. One major new aspect that MassIVeQuant brings to the table is that there are new dataset formats where new collections are added to the dataset for quantitative information. So there's now a way to add metadata to a dataset where you actually can describe the samples and the experimental design for the dataset. There's a way to submit quantification results in formats that are reusable, and there's a collection of formats that are supported. And there's a way to support the results of the statistical analysis of quantified analytes. There's a collection specifically for that. The metadata that is submitted is in a very simple format, which is modeled after the MS Stats input format, which you probably already heard about in the May Institute, because our focus was really on capturing the metadata that is most practical and usable for the data set. And in this case, the most obvious thing you'll want to do is run some kind of differential analysis in your data set. And for that, what you need is the input data that you pass to MS Stats. So let me see here. There's a question, can be difficult to get an identification file for DIA data. How do we create a complete submission for DIA data sets? So this is a good question. How do we do this for DIA data? So we actually have been able to do this in two different ways. One is it is possible to create an MZTab file or an MZID file that has the identified peptide or precursor, to be more accurate, and then links to a scan in a DIA file. So our tool, the mSplit DIA that we use to process DIA data, actually outputs that kind of format. And the current situation is just that most tools for analysis of DIA data do not output anything like that. But even though they could. So I understand that for people who are using the tools, it's a challenge because the tools don't support it and you just don't have a way to do it. But I would ask the tool developers for the programs that you work with, that ask them why can't they do it? Because they could. And if they could do that, the data could be submitted as complete. Another approach or intermediate approach that has now been also supported is with DIA Umpire. When the data is processed, they export the MS2 spectra into a pseudo MS2 file. And those files can then be searched as regular data dependent files. And the files that you get with the IDs as a result can be used to create complete submissions also. So it's not perfect because ideally we'd like the IDs to go all the way back to the DIA file, not just to a processed pseudo MS2 file, but it's better than nothing. So from that perspective, it also gets you a foot in the door, although definitely the preferable solution would be to have the tools that process the DIA file actually output at least one representative scan for each precursor that tells you maybe the peak of the elution profile, if not all the scans where the precursor is detected. So it's not a question of file formats, it's a question of support from the tools. And hopefully we'll get there in the not too distant future. All right. So the third major aspect that MassIVEquant has contributed to the conversation is that we now have executable workflows directly in the system. So you can actually run MS stats directly at MassIVE. And if you already have the quantitative results extracted from the data, if you don't, MaxQuant will soon be available as a workflow that you can use to process any public data set or your own privately uploaded data, and you can run it online, and then pass it to MS Stats to do the differential abundance analysis. Once you have several results available, you can do results comparison also at the quantitative level, and we'll probably have time to hear about some of that today. And there are interactive results views in the system to allow you to interact with the data. So let me see if I can move this forward. There you go. So when you get your MS Stats results, they're shown in a table that you can filter and sort by. You can filter by p-value, sort by full change. And at the click of a button, you can see the columns expand, and you can see the differential abundance as it was measured in the different samples, in the different conditions, and the different replicates inside each condition, as well as the full change. So again, all these things very conveniently available just by clicking on buttons on an interface. And that's really where the session today will go into, is all the sort of how you can access all of these things in the system. And just before I close, I wanted to very quickly just mention a new project that we've also developed at MassIVE, and that is directed to responding to this pandemic that is causing all of us to have to attend this session from home instead of being all at the same place and having beers after this is done. But as it is, because of this pandemic, many things have changed across the world. And one thing that we thought should also change is the speed at which we share mass spectrometry data for data sets that are related to coronaviruses, and in particular to SARS-CoV-2, the virus causing this COVID-19 pandemic that we're all in. So we've created a new resource to expedite the sharing of this data and the reanalysis of this data. And by allowing us to have all of these things in the same place, that we are able to compare the data, inspect the data, and hopefully move faster towards understanding what it all means. So I don't have time to go into details for this resource, but I just wanted to mention that it is available, and if that you're working on projects related to coronaviruses, would be glad to hear from you and let you know more about how to connect things to this. And with that, I would stop here and be glad to answer any other questions if we have time and there are any,
So
or
we
we could get
can take
into
a
the
couple
next session.
minutes if there are questions. But otherwise, our next step is for Meena to actually do a demo of MassIVE.quant and look a little bit more in practice what it means to actually upload the data or reanalyze the data. And this includes submitting the data sets themselves, but also submitting reanalysis of the data sets using whatever workflow you personally favor. So I think we have a couple of questions, Nuno, if you don't mind.
Excellent. All right, so let me go through these. Does it contain metabolomics data? Yes, that's the short answer. MassIVE supports all kinds of mass spectrometry data. And the primary portal for metabolomics data is our GNPS resource. But all of GNPS is built on MassIVE, just like MassIVeQuant and just like this Corona MassKB. So the underlying infrastructure is all MassIVE. So there are thousands of metabolomics data sets at MassIVE, and would be glad to get more. So in particular for this, even here for Corona MassKB, there's already metabolomics data sets available, both for SARS-CoV-2 and for some of these other viruses. So definitely the answer is yes. There's support and interest in the metabolomics data for sure. The second question, which is, can we get a PRIDE ID if we submit to MassIVE? Note that what you need for publication is a proteomics change ID, and things have gotten a bit confusing maybe because the identification that PRIDE uses is the data set identifier they use at PRIDE is the same as the proteomics change identifier, but what you really need is a proteomics change identifier. It's just that PXD followed by a number. And the answer is absolutely yes. You can get that at MassIVE, and you can get that at any proteomics change repository. So as long as you're a part of the consortium, you will get the PXD identifier. As long as the repository is a part of the consortium, you'll get the PXD identifier that you can use for publications. So absolutely yes, you can definitely get it if you do it at MassIVE. And I would also like to mention that if you're interested in reanalyzing data that was submitted to to PRIDE or to the other proteome exchange repositories, but definitely with PRIDE, we can automatically import the data into MassIVE, so you can do the reanalysis and you can do the comparisons, and you can add it to MassIVeQuant. A good number of these data sets that we have here in MassIVeQuant today, several of them were deposited initially at PRIDE, but they're now also at MassIVE. And it's the same data set, it's all the same data, same identifiers, but now you can reanalyze it here. So short answer is yes, we can get the proteome exchange identifier, which is really the one you want if you submit to MassIVE. And even if it wasn't originally deposited here, we can get it here to do the reanalyses. So great to share proteomics data. However, for quantitative analysis, what are the strategies about QCing the uploaded data, not knowing the experimental designs? This is a great question. So there are tools that can be used to do this analysis. We are starting to develop reports to generate QCML files as a result of the data that's uploaded. That's still work in progress at this stage. But the way that we learn about it is when running the reanalysis and seeing what results we get. One of the tools that we use at the start for pretty much all uploaded data sets is Paramedic, which was developed at University of Washington and Bill Noble's group. And we collaborated with them on that development. And we essentially use it at the start of every experiment to learn about mass tolerance, mass errors, and potential PTMs in a data set and things like that. For example, it automatically tells us whether a data set is TMT or not, regardless of what people wrote in the metadata for the data set. But then to know the experimental design and the sample metadata, that indeed has to be submitted with the data set, the description, or it can be added later. So some of the data sets here in MassIVE.quant were originally submitted without any metadata, but we were able to go back to the paper, figure out what that metadata was, add it to the data set, and now use it for the new analyses that are shared. So those analyses share and contribute not just new results, but also the metadata, the structure of the experiments, which you can then use to do your own reanalysis going forward. So a good number of these things will be apparent today as we go through the demo of how to use the system. Can it reanalyze TMT data? Absolutely, yes. It can reanalyze TMT data both for the analysis of modifications and searches and for the quantitative analysis that follows. We have already workflows at MassIVE that extract the TMT channels abundances from the identified precursors and provide those in a format that can immediately pass onto the MStats workflow to do the TMT analysis. So it really streamlines this process. I believe today we're already going to start with cases where the TMT data had been extracted previously, but we now have ways to do that directly in the system, and we should be able to show that soon. But it's definitely possible to do this both for ID and for the quant analysis.
No, in the interest of time, I think we should probably leave the other questions because I think many of them will be clear during the demo.
Sounds good. I'll also respond to them in text. It's easier.
Exactly. So if we do have some questions which go over time, we will certainly save them and post them in the Google Doc, which is associated with this session. And we will make sure to provide answers in writing after that. Well, Nuno, thank you so very much. So our next presentation is from Meena, who will now do the demo of MassIVEquant. So if you are joining us for the first time, Meena is an associate research scientist in the Curie College of Computer Science at Northeastern University. Meena is the lead developer of MStats, and she was also leading our collaboration with MassIVE to integrate quantitative data and reanalysis of quantitative data within MassIVE. So I'm confident that many of these questions that are posted here, Meena will be able to answer. So thank you very much, Meena.
Thank you. Good morning, good evening, good afternoon, everyone. So I'm back. Today I will just demo or how to use the MassIVEquant. So we have a slide. This slide also, there's a link for this slide in our Google Doc. So, let's start. So the Nuno already go through all the details of what is the new feature or something, but I will just go over one by one. So first, you can go to the MassIVEquant webpage here. There's a link if you can find it. The easy way is just in the MassIVE website at the bottom. You can see here MassIVEquant, and then this one, and then you can see this website. So here is all we have for MassIVEquant. So here it showed us some statistics, show how many public data is available for MassIVEquant, and then how many the quant reanalysis available, and then number of the data set by species over here. So we can browse the public data in the MassIVEquant, and then we can submit the reanalysis, and then we can access the reanalysis, and then we can run the MStat workflow, and then we can run the comparison workflow. So we will go over one by one. So let's see the first one. Before we go to the first one, if you want to use the fully MassIVEquant, you need a two-step. So first, you need to register account in MassIVE. So here's the documentation and video how you register account. It's very simple. So basically here, you can just click on this register button, and then you can just put your information. And then you need to upload your data to your account in MassIVE. So which means you have a bunch of the data file in your local computer. So to make this available to MassIVE, you have to copy this file to your space in MassIVE. So after you register your account, you're gonna have your space in MassIVE. First, you have to copy this one, use the FTP. So there's also documentation and video out there. So then let's see the browser public data set. Now we have a 42 public data set, which include the 10 data set, 10 control mixture, 3 DDA, 4 DIA, 1 SRM, and 2 TMT control mixture data. And then also 30 the biological investigation. There are a bunch of the stuff. So how we recognize this data set in the MassIVE.quant? You have raw file for that experiment. You have quantification data. You have metadata. And then if you have the statistical analysis, then the your data will be added to the MassIVE.quant. So let's see. First, go to the browser data set. So here it showed the 42 data set. So here's for each data set, and then here's the all information we can get. So this is the data set, and then we have a MassIVE ID for this data set. And then we also have Proteome Discover ID. And then here you can see here's the plus button. If you click the plus button, it said this data set have quantification result, differential abundance result, and the study data set, the study design. Study design means you have a metadata. If you already have this information, the MassIVEquant count how many conditions, how many biological replicate, and how many technical replicate available in this data set. And then also show how many proteins are quantified, and then how many differential abundance proteins available for this result. And also, you can see who actually can go, wanna go right here. Also show the originally this data set from the which lab, and then what species they use, and then also show how many quantification reanalysis available, and then where it is. So I will talk about what is the data, what is the container, and reanalysis. And then if you scroll down here, sometimes you can see this plus button. There are 3 reanalysis container, and then they have 3 different analysis, sometimes much more. And here you can also see the updated the data and so on. So basically you can search the data set which have all the quantification data, and then metadata, and then all the details. So that is a brief overview about how we browse the data set. Then let's move on to how to submit your reanalysis. So we actually came from this idea to store all the intermediate data. The first, actually, I have a difficulty to find all the information, the quantification. For example, I'm a statistician. We want to develop the model, and then we want to test our model for a variety of the data set. So, we have a really good repository to save the thousands of data set. So we go to the MassIVE and then Pride, and then to reanalyze everything, we actually need some intermediate data. However, most likely, most data set, they have only raw file data set. And then all the intermediate is really hard to find it. Even the experiment design, we cannot find what exactly look like. Then we actually cannot reproduce all the widget. So, okay, so let's say, okay, we have a raw file. We can run everything. So we can use in any kind of data processing. It generates the multiple intermediate files. Okay, we have a whole intermediate file for one analysis. Then, how can I save this one? How can we make this available to the public? So most likely, nowadays many people use the GitHub. So GitHub also, we can put all the intermediate file. However, there are some limitations. So first one is the data set, the size of the data set. They have a limit of the size of data set. Sometimes if a really big one, we cannot do this. And basically raw file, we cannot save the raw file in the GitHub, which means raw file or some files are available in the Pride or MassIVEquant, but some of the file is available in the GitHub. And then this GitHub means for each person's account and so on. So it's really hard to navigate to where is it, and we can make a link, but this part is over there, this part is over there. It's quite confusing. So I want to put all the reanalysis in one places, and then it's really easy to find all the intermediate file. So as Nuno mentioned, the MassIVE, now they have the MassIVE made a new category for quantification. So for example, quantification widget section category. So we can put any quantification widget in this category. And then we have a metadata section, which include the experiment design. And then we also have a section for statistical analysis section, which means we can also put the test, the widget one. So it's really easy to navigate which file should be where, and then where we can find the specific output. So here is the basic concept and terminology we used in the MassIVE. So there are three hierarchy structures. So one is we call data set. So this is a kind of the MassIVE ID, MSB, and XXX. It's the same as the Pride ID, the protein exchange ID. So it's just kind of same ID. So it could be experiment level or publication level. We can see many publications, they just put all the data set in the one repository, and they have a one PXD ID. And then we say, okay, you can find everything over here. That is kind of, we call this is data set. Under the data set, we can make a container. So container, so like the subspace in the data sets. So it can be experiment or set of analysis, which means, okay, I want to set my container under this data set, and then I will put all the reanalysis in that specific container. And another researcher, they can make their container under the same data set, but it will have a unique container ID. So here, a container ID should be R, MSB, and XXX. So a container can have one reanalysis, or it can include the multiple reanalysis one. And then the bottom one is we call reanalysis. It's basically the result of the reanalysis from the raw file or very first one. And then it will have RMSB XXX dot X. So we have these three layers in the MassIVE. So how it works. So for example, we have many steps to analyze and to get the final conclusion one. So we have some sample preparation and then run the mass spec equipment. So let's say this is the one experiment, and then this is another experiment and another experiment. And then we can use some different data acquisitions. And then we are going to do the identification using many or one or many combination of identification tools. And then also for quantification, there are several data processing tools, so you can choose anything. So for example, here, one data set at the first, we use these three searching algorithm, and then use a skyline, and then use also some option for statistical analysis, and then we have the one conclusion. Next, we use the same raw file, but maybe we use the different searching engine and different the processing tool, and then do also some different statistical analysis. And then we're gonna do the different result. So each result, all like this one pass will be, we call this is a reanalysis. So you can make any combination of this, and then you can generate the like large of the reanalysis. So we can save each analysis for all the intermediate file. You can set it up for each analysis. So here's the example. So here I will go to this one. You can go to the link here, but I want to show how we can search in here. So if I know the MassIVE ID, let's say 79843, you can search here. So here's 79843. And then if you click filter, it will show. So I found the example data set over here. This is the IPRD data. If you attended yesterday, this is the one of the control research data. We did a demonstration. So this MSV, so like really top level, data set level, it has 57 reanalysis. And then here you can see this specific cases. You can see this is there are A condition, and then 4, 3, and then hold a summary of this. And then bottom here, you can see the list of the reanalysis. So it showed one row showed the one reanalysis. So it have a title, and then it also showed this specific reanalysis belonging to which container. So for example, here 249 reanalysis container, they have like large of the reanalysis there. And then there's also another one, the container is the 248. They also have a lot of the reanalysis. So how this one is kind of organized is, okay, this case for this data set, we have a one experiment, and then we have a one, the data set ID. And then over here, we actually have several data sets, but this is two container what I made it. So one is a 249, the other one is a 259. So for example, here case from the same data set, same raw file, I just run the same raw file with the different processing tools. So for example, this reanalysis, I just use a Skyline, and then I just go through the MSDAT, and then here's the result. So it includes a Skyline file. It includes the quantification from Skyline, and it includes the metadata. It includes R script, what I use for MSDAT, and then it includes the statistical analysis result from MSDAT, and then also include the log file, what the MSDAT generated. And then I just repeat the same thing with the MassIVeQuant. For MassIVeQuant, yes, I just go through the same workflow, but it has the output of the MassIVeQuant. It should be the parameter file, evidence.txt, or the protein group file, and then also include the R script and then statistical analysis by MSDAT and log file. And then I just repeat this one for the proteomics. So this container, I just use the different processing tools. And then I set up another container. Okay, I have some result for Skyline, but I want to change some option in the MSDAT. So for example, I use the Skyline for quantification, but for MSDAT size, I just change the one parameter for MSDAT. I just use the top 3 feature, not use all features. Then I just go through everything, and then we have a result. And then I just put this one as one analysis. This is a real analysis with a different other parameter setting. Then I also repeat the same thing. Next, I just change again. This one I use the top 5. At each step, final conclusion will be different. So I just save this one is kind of the one other, the branch over there. So for example, here I can compare how it look like with the same MSDAT setting, but different processing tool with a different quantification. Here I can compare with the same quantification from Skyline, but with a different option for MSDAT. So it is up to you how you can just compare this, how you set up the container. But main point is I can say, if I say, okay, I compare this, And then if you go to this specific container, you can see how how much we have a different quantification widget. Or I just point and give this container ID. Okay, here you can compare the MSDAT of the different option. The second one is also another different way how we organize these branch structures. So for example, I want to check this one. So 84864. I will check. We'll go back to our browser one. Okay, so go back. Here I said 9 numbers. 84864. I search it. Okay, here's a data set. So I click this one. Okay, here, this is our MassIVE ID. And then you can see this is the partial, which means no identification widget one. This is a public, and then you can also see that this is the proteome exchange ID. So I just click this one. So this one originally submitted to the PRIDE. And then here's how you can see it. So if you go to the FTP4 location, it will show all the files under this PRIDE. Here, everything is there. Everything is there. Raw file, lots of raw file, lots of TXT, SNN, and so on. So when I see this, oh my God, so many files. So what each file means. Definitely I can guess this is a raw file, right? And then this is a MassIVequant. Okay, so good enough. Problem is this data set, this is for one publication. For this publication, they actually use several different experiments. So this is a combination of the different experiment. So they just do the one set, that's several set of this. So it's really hard to navigate, okay, which file belonging to which experiment, and then kind of things. Also, and then here we can guess this is a raw file or something, but it's not marked. But the PRIDE needs to support to show which file for which step. So for example, here, there's a type. So this one is a type raw file type. And then here, okay, for example, if not very specific case, they put these others and others. But still, it's not easy to find the experiment. So how we can set it up for here, we just import this the data set into the MassIVE and then kind of reorganized. So what I did, I just go through the publication, and then I can organize, okay, this one should be here and here and here, and then also talk to the authors and then confirm this one is okay. So as I mentioned, this publication, they have the 3 experiments there. So I set up the container for each experiment. So this container have this specific experiment, and then other container is for this specific container. So each container belonging to the raw files for that specific experiment. And then the other container also include the raw file and every an intermediate file for this specific experiment. And then use this one. We also set up the reanalysis. So let's go back to the original here. So for this data set, this is just a more whole reanalysis one. So now you can see there are 5. Here you can see this is the container for this specific data set. This is 280. And then 279, this container include this specific experiment. And then 281 have also this different one. So let's click one of this. And then the container have all information for this specific experiment. It has only one reanalysis. It has a 10 condition and 3 biological replicate. And then here we go. And then if you go click the PDF or download, this is what I submitted over there. And then you can see here's the subfolders. So metadata, you should find the experiment design, how to annotate it. If you click it, you can download it. And then for quant part, if you click it, it will include the quantification, so report from SpectralNet. And then here, the quant stat one should include the statistical analysis one. So, and also here, the method section, it includes all the information about the method, especially R script or the description of the PDF and so on. So basically, if you download this R file, if you use this quantification and then just run it, you should have the same result for this quantification, the statistical analysis result, if you have a same version or something like this. So that is kind of the main idea. So, and then I will check another container, so which include a different experiment design. So now you can see the experiment design numbers are different than before. So previously, I remember it's a 10 condition, but this is a 3 conditions there, and then there are 12 biological replicates. So easy to look at what is exactly this experiment look like, and then what, you can download all the intermediate file. So that is the main idea for submit reanalysis. So, okay, so time goes so fast. So I will show quickly how you can do it. However, this is how you can do it. Click the for each step, we have a documentation in the video tutorial. So for example, click the documentations. You can see for that specific step, you can just follow this. Documentation. You can see what kind of the screenshot you can see. And also video tutorial, each one should be not that long. The maximum is 10 minutes. So video tutorial, we already have a YouTube, like so our lab, the YouTube channel. So you can just follow what I did, exactly what I did. So for example, here, so I made the quantification. Yeah, here I just do the demo for this one. So if I couldn't finish all of them, you can also find this one. And then I use the example data from the MassIVEquant itself, so you can definitely do this. But I will show the shortly how it look like. And so let's go back to our IPRG data, which we use a lot for training purpose too. So what was the number of, so I think 79846. So I have a question. So should the different container have a same raw file? It can be, or it doesn't need to be. So if I use the same raw file, but I just set up the container differently, yes, definitely you can do this. Or you can, if what I mentioned, the second case is if they have a multiple experiment design, you can set up the container with a different raw file. And actually, you don't need to save raw file every time, because the data set structure already have a raw file. So basically, container we can just point out I use this raw file. So you don't need to upload the raw file again and so on. You can just point out I used this specific raw file in our data set. So here, IPRG data, let's go. So this is the public data set. So if you, I just show how you can add the reanalysis for public data set. So definitely if that is private, if you are not owner for that data set, you cannot see it. If you are the owner for private data, you can also add the reanalysis for your private data set. But here is already, I just want to show that the public one. And even if your data is a private, also it will not show up in the MassIVEquant. So here there are several things. Okay, here click add reanalysis. So it takes us some. Okay, so here you can see I need to log in first. So if you need to submit the reanalysis, you need to log in first. Okay, so I will search again 79843, right? Here. reanalysis again and add reanalysis. So it will take, it's enough reading, it's working. So now it said add the files to massive data set, but it will not change it. It takes some, yeah, here it goes, it's working, right? Here they go, it automatically select the massive data set, add reanalysis widget. So if you click this, actually there are several workflows there. So I can show you something, but this is how you can use the MSDAT workflow. And then here's how you can use the MSDAT comparison. But here now, just add the reanalysis here, and then you should type the title for your analysis. So it would be good to make distinguishable for other reanalysis, and it should be more than 30 characters. Let's say just test. And then this one, the species instrument, this is already imported from the original data set. You don't need to select this. Then here is in the most important section, you have to select it. So here there are many categories. If your analysis more focused on identification, you can also select your new identification widget and then submit it. But here we are focused on more quantification widget. So we are gonna submit the quantification, metadata, and statistical reanalysis to add to the MassIVEquant. So click any select input button. And then select input file means we have to select files in your MassIVEquant. So that's why you have to copy your file from the local computer to the MassIVEquant first, and then we can select the imported file. So for example, here's the categories. So, and then it will show all the files available in my MassIVEquant. I know this is really a lot, but here, this is, here's also the one tip is, here's a data set. It showed all the data set while you select it. And then here's my account name. And then it showed the files available in my account. So it's under the Meena Choi. They are, okay, here, the IPRG data set. And then here you can see, oh, this one probably the wrong one. Yeah, I tested several times. So, okay, so this one. So you can find my file, what I uploaded. So for example, here, this is annotation. So I have to click this annotation, click. Then you have to select this one should go to where? It should go to the metadata. Click metadata, and then you can see this is selected to these categories. And then here, I have R file, and then description, and then this log file. This is related to the method. And then here you have to click the method section, and then it will show up. So after you select everything, you have to click the finish select button, and then it will show. Then next step, you have to select which container should be this analysis should go. So here now we have several container. If you want to submit this reanalysis to some specific container, you can select it. If not, you can make a new container, and then you can set up the titles.
Meena, could you take one question? And also in the interest of time, probably we should answer the remaining questions in writing after that.
Right. Is it possible reanalysis of several data set together? So this is depending on what kind of reanalysis you mean. So, so I think you mean the several data set means some different MSB ID. That part I'm not 100% sure. I'm not 100% sure, especially for identification one. But the quant part, probably I will mention this one again. So I will go through quickly. So, and then if you fill this one, you can submit and then click submit, and then we're gonna go over. So that is how you can use this workflow in the MassIVeQuant. So as I shortly mentioned, and then you can access each analysis, for go to the each analysis, you can see a summary of each analysis. And plus, if you click the FTP, you can download all the files. And then, as I briefly mentioned, MSDAT workflow, you can select here, MSDAT, and then it will take a few seconds to change everything. Yeah, because I already started to do something, they are not recognize this one, but it's the same thing. Here, go to run MSDAT workflow. It will show the MSDAT workflow over there. Here, you have to select the input file. To run the MSDAT workflow means for MSDAT, you need the quantification result, you need the annotation file. So you have to put your file into your MassIVE account, or you can you can use another reanalysis output over there. So here's how you can use the MSDAT workflow. So for example, this is the one MassIVE data set. So they have a raw file and FASTA file. They don't have any quantification one. Then you can download, or you can just use the MassIVE to run identification and quantification. So download all file to your local computer and then run this processing. And then probably they generate all the quantification one. Then you can submit this quantification result to your repository, and then use this MSDAT workflow, and then they're gonna generate all the output, and then you can just select the result and then submit automatically in the MassIVeQuant under this data set. Or you can just use already published or publicly available for quantification data. Just use the MSDAT workflow in MassIVE, and then you can just submit it, and then you can rename it as a different reanalysis. Also, what other thing is possible is, okay, there are a bunch of the reanalysis available over there. Then if everything is just one spaces in the branch structure, it is easy to compare their result if they have a quite consistent columns informations. So for example, this case, I processed the several file using the different processing tools. And then there are the same format because I use the MSDAT, but the conclusion will be different. Then we can actually compare this one in the MassIVeQuant. So here, if you go back here you can click run workflows. Here, MSDAT comparison. So this one we call it MSDAT comparison because this workflow is very specific for the columns for MSDAT. So here you need to select which file, which statistical analysis result you want to compare from your repository or some other repository, and then you can compare this. So how you can select your files and so on, it is available over here. So that is the overview for this one. And then again, all this single step how you can select where you can find or some very small tips is available in the documentation and the video tutorial. So I will answer some of the questions. Okay, if I want to reanalyze someone else's data, do I have to download their data set and upload it in the personal folder in the MassIVeQuant? So depending on what kind of reanalysis you want. If some automatic workflow is available in the MassIVe, you can directly use it. So for example, I just want to, but if you want to reanalyze your data set for your purpose using some different tools or your method, definitely it should be downloaded for your computer, and then you run it, and then you can submit this result for reanalysis like this. Okay.
Okay, thank you so much, Meena. I hope that this was useful, and I hope that all of you who generate the data or process the data or develop new methods for analysis of data will make use of this resource. So this is definitely useful for you to have data to benchmark your methods or have multiple tools to compare the analysis results of the data sets you are interested in. And we already have enough examples of controlled mixtures where you have some notion of ground truth, or biological investigations with different types of acquisition workflows. So we hope that you will use it, but we also hope that you will actually contribute new data sets and new reanalysis. So currently, all the annotations of experimental design are very heavily manually curated. So all the new submissions to the repository are double-checked essentially by our two groups. And in the future, as Nuno mentioned, we will be looking for some automated ways of doing this. But as of right now, every single data set which is in this repository was very heavily checked manually for consistency.